Hi everyone, this is Jayita Goswami. I'm a molecular biologist and biochemist and taking part in our virtual ASBMB meeting. I was supposed to present this poster and talk drug delivery methods, application and new horizon of conjugated drugs. As you know that our life and drug is inevitable. We always face various diseases and that has to be addressed by various drugs. Sometimes some drugs fails. We already tried mist, intravenous, and also oral in various different ways. Now it's a field of conjugated drugs. We have used DNA, RNA, protein, nanoparticles, conjugated new drugs already. Now researchers are thinking about a lot more varieties in it. And I worked with cancer, diabetes, microbiology projects and also other plant related projects too. So when I was addressing my cancer project, I used various, actually 200 rare peptides. Out of 200 I got only few that was active against the cancer cells. I worked with 8 different cancer cell lines and it was a very rigorous and tedious work. And majority of those were rare peptides. It had solubility issue. So when I added an arginine tail with my peptide, it increased the solubility and also helped with the cell penetration. For the experimental method, I used various proceedings. One of them was obviously particle bombardment, microinjection, cell culture, and also nanoparticle conjugated drugs to see the peptides co-localization. And also I used confocal microscopy, laser confocal microscopy to have the high resolution picture of my culture cells and also the co-localization. I also did the dose curve to see the IC50 as well as I studied the carrier vehicle very nicely, because any drug, when we are dealing with drugs that has some shelf life matter issues, like sometimes it crashes in certain media and sometimes it even crashes in the fridge too. So I had to deal with those. And here I came up with some of my new idea about the drugs. Carbon nanotube antibody conjugated with vitamins for the cancer therapy, and also bacteriophage when it is the genetic material is completely removed from the bacteriophage. So, and also fullerene natural dendrimers are also a good source of alternative conjugated drug sources that we can use to deliver our drugs in the cells up to subcellular organelles. And I find mitochondria is a very interesting subcellular organelle where we see a lot of damages can happen because of inflammation, and also genetical disorder can happen, and the genetical disorder also related to mitochondria. And obviously mitochondria is the power of the cell. Without power we can't do anything. So if we want to deliver a drug in a cell, mitochondria is a good target to deal with because it takes care of a lot of things. And when I was working with the diabetes project, I was sequencing and analyzing various genes motifs that is related to the diabetes. So diabetes eventually can give you cancer too, because diabetes is such a slow killer. Eventually it creates cancer as well. So I found that a particular gene can be in several different chromosomes. It's not just in one place. So if you are planning to knock down one gene, it cannot be knocked down all the way, because there are several genes, actually millions of genes that carry copies in different chromosomes. So if you knock down just one, it does not silence all the way, and all those genes sitting in other chromosomes, they're associated with other biosynthetic pathways that are also important for your cellular activities. So nowadays we are in the drug front. We are thinking about more innovative, brilliant idea about making fullerene rendimers, natural rendimers, hydrocarbon flask as a drug delivery vehicle, where fullerene is a very fascinating carbon-carbon bond C60C120 carbon compound, where you can put your drug of interest in the middle, and then you can deliver it in the cells in vitro or later on in vivo. And hydrocarbon fuzz is also a brilliant idea, although these molecular designs are very complex. You can do it in vitro in the laboratory softwares sometime. A lot of time actually it also fails in the laboratory when you are running the experiments, because it's always, research is always trial and error method. So you have to come up with various designs. Finally you will get one successful candidate. And natural polymers are very interesting, like the chitin, chitosan, other cellular moieties that we get on the surface of the cells, bacterial cells or the viral cells. Those are also a good target where we can find natural co-bonding, like always it works like a ball socket kind of reactions. All this natural, your drug of interest and your binding moiety should be very close, and also our body has tendency to pick up something closer to the body's own cellular components or the own structure. So that's how we can use the natural carbohydrate complex hydrocarbons as a drug delivery methods. With this, I would like to conclude my talk today, because I was finding hard time to deliver a big speech that I planned before. I would like to acknowledge and thank NIH, DARPA, ASU, BSU and other companies who gave me opportunity to work with them. Thank you.